first one is Dr. Dylan Murray, and the second one is Dr. Henry Paulson. Each presentation will take about 30 minutes, followed by 10 minutes of Q&As, which you can post in a Q&A portion of the Zoom window, please, or you can post it in your chat. So the first speaker is Dr. Dylan Murray, who is an assistant professor at UC Davis. Dr. Murray did his PhD thesis in the lab of Dr. Tim Cross at Florida State University at Tallahassee. And then he joined the lab of Dr. Rob Tico for his postdoctoral studies. And after spending 4 years doing his postdoctoral research, he joined UC Davis at 2018. So it's quite recent. And the focus of his research is on understanding the physical chemistry of low sequence complexity protein domains and how these properties relate to their microscopic behavior in the cells. Welcome and thank you very much for coming today. It's your turn.
Okay, well, thank you very much. I really am very grateful for the opportunity to tell you about the work that my team at UC Davis in the chemistry department has been trying to do to understand low complexity protein domains. Okay, so first I'd like to just give you an overview of what I'm going to talk about today. First, I'm going to tell you about why low complexity protein domains are interesting. And I really wanted to point out two pieces of work that have come out of my lab. First was published in JACS about a year ago about TDP43. And this other work, which we actually posted to the bioRxiv preprint server last week. So this is very recent work related to the protein TIA1. So last summer, Google's DeepMind released their artificial intelligence algorithm looking at protein folding, right? So as many of you probably know, this has been a substantial problem in protein chemistry over the past 60 years or so. And that's where we need to take this single letter amino acid code and try to fold it up into some three-dimensional structure. And they got very, very good at this. And they got so good at this, they did this for every protein in the human proteome, as well as some other organisms. So this is just one example of a prediction from the DeepMind database. On the left is the domain structure of the TDP43 protein. It's got some N-terminal domains and a C-terminal low complexity domain. So if you look at the structure here on the right, you can see that there's three well-folded domains with a high degree of structural accuracy. And these correspond to the N-terminal domains. What's interesting about this is that the C-terminal low complexity domain, which is this orange region lacking any structure in this prediction, is not predicted to have structure. And I find that particularly interesting for a reason I'll get to in a moment. And when they released the database of protein structures, I got really excited because I immediately went to the database and looked up every protein that we study in my lab, which all of them have a low complexity domain. And what did I find is that each one of them has a low complexity domain that's not predicted to have structure. And I find that interesting because that means I have a lot of work to do. So one protein that I've studied extensively with the low complexity domain is the fused in sarcoma protein with the prediction shown here on the left. And its low complexity domain is this N-terminal 214 residue sequence folds up into amyloid-like fibril structures. We can look at these under electron microscopy. They form the sort of the stereotypical fibril shapes. We have high resolution structures of these protein monomers and these protein fibrils. So we know that low complexity domains are capable of adapting structure. It's just that there's something different about them that does not permit the DeepMind algorithm to predict their three-dimensional structure. So shown here is just an example of a low complexity protein domain. It's the TDP43 C-terminal domain in single letter amino acid code. So it's a low complexity sequence. That means it's biased towards a subset of the 20 naturally occurring amino acids. In this case, glycine, serine, asparagine, and alanine make up 64% of this protein sequence. And it's interesting to compare this to other protein sequences. So if you look over here on the histograms on the left side of the slide, you can see that for T4 lysozyme, which is just a sort of a canonical enzymatic globular type protein, you can see that it's got a relatively even distribution of polar, hydrophobic, and charged amino acids with relatively few glycines. If you compare that to alpha-synuclein, which you might think of as a purely pathogenic amyloid, you can see that it's got a relatively similar distribution of amino acids. But the low complexity domains, the sequences here on the right of these histograms, are highly enriched in polar and glycine amino acids. The fused in sarcoma protein has relatively few hydrophobic and charged amino acids, whereas the TDP-43 has slightly higher content of hydrophobic amino acids. Okay, so now that I've introduced the low complexity protein to you, let's talk about the TDP-43 protein. So the TDP-43 protein, as I kind of hinted to earlier, is an RNA binding protein with a low complexity domain. In this cartoon of various aspects of RNA metabolism inside a cell here on the right side of the slide, the TDP-43 protein is these orange squiggly lines here. So you can see that's involved in a large number of biological processes, mostly focused around RNA metabolism. And what I'm going to focus on today is the conversion of stress granule structures into pathological aggregates. So the full length as well as C-terminal truncations of the TDP-43 protein are found to aggregate in patients who have frontotemporal dementia and amyotrophic lateral sclerosis. And in fact, 49 of the 56 or so disease mutations that are linked to ALS all reside in the C-terminal low complexity domain. And on top of that, residues 311 to 360 have been a particular point of focus because there are some assays that show that this is the most aggregation prone part of the protein, both in cell culture as well as some purified protein work. So it's really exciting to me as a molecular biophysicist that we can reproduce the assembly process of the stress granules to the pathological aggregates in the test tube. And we can do that by preparing denatured states of proteins, letting them assemble into these liquid droplet-like structures under controlled conditions, and then allowing them to age, where we tend to get fiber-like structures as these droplets age and mature. So before starting this work, there was some work, information known about the TDP-43 protein, in particular work on residues 311 to 360. We learned that it undergoes a helix to fibril transition, which is what I'm showing you here. So the solution NMR structure of the helix is here on the left. Under certain conditions, it will form this fibril-like structure shown here on the right, and you're looking down the length of the fibril in this particular model here. So the fact that a subset or a sub-region of a low complexity sequence forms fibril-like regions is not unheard of. We have the HNRMPA2 protein here on the left and the fused in sarcoma protein here on the right. And in both of these cases, they fold up into regularly well-defined protein structures that compose roughly 20 to 30% of the entire low complexity domain sequence. So what students in my lab did is they took the complete low complexity domain. So this includes the aggregation core as well as N and C terminal residues flanking this core. They purified it to homogeneity in denaturant. We can remove this denaturant, get beautiful looking liquid droplets, and then allow those to age into fibrils and use solid state NMR to understand the structural process. So this is just a bright field image of some of the droplets that we formed. I'm not going to show you the video, but we did observe multiple instances of droplet fusion where two droplets come together and create a larger droplet, which is one of the signatures of a liquid-liquid phase separation. We don't have fluorescence molecules attached to our protein here, so we're unable to do the sort of fundamental FRAP experiments that folks like to do. So anyway, we took these droplets and we just allowed them to age in the test tube. And as you can see with these bright field microscope images here on the left, they gradually form these amorphous looking aggregate structures. Turning to a slightly higher resolution technique, we use transmission electron microscopy, and these negatively stained electron micrographs show that amorphous aggregates appear as the droplets disappear. They transition into heterogeneous fibril-looking structures that then mature into these highly bonded fibers over the span of a week. We did fluorescence assays to look first at thioflavin T, which reports on the formation of cross-beta type structures. And we observed the typical or the expected increase in thioflavin T fluorescence as the fibril structures form over time. We also looked at tryptophan residues, and we observed a decrease in fluorescence and a shift to lower wavelengths, which is consistent with some of the tryptophans removing or losing their ability or their access to solvent, but the location of the tryptophan residues in the protein primary sequence do not allow us to make any distinct conclusions based on this data. So we turn to the solid state nuclear magnetic resonance. Shown here is a spectrum of the TDP-43 fibrils as the droplets age, or after the droplets have aged, excuse me. These were cross-polarization solid state NMR spectra, so they report on the highly immobilized regions of the protein fibril. These carbon and carbon-nitrogen spectra, the signals observed in these experiments arise from magnetization transfers that I've diagrammed for you here in the middle of the slide. Based on the distribution of amino acid chemical shifts, we can immediately identify regions of interest in this spectra or these spectra. For example, serine, glycine, and alanine residues are very prevalent in these fibrils, whereas glutamine and methionine residues are essentially absent from the nitrogen-carbon spectra and very weak in the carbon-carbon spectra. So if we compare this to the amino acid distribution of the low complexity domain, we can compare the signals that we observe in the age droplets to the solid state NMR study from Junqian Liu and co-workers of the aggregation core by itself. So as we expect, we see lots of glycine and serine residues, both in the age droplets as well as in the aggregation core solid state NMR study. But what's particularly interesting is that we do not see any of the glutamine and methionine residues that are so prevalent in the aggregation core. And that immediately tells us that we're looking at a fibril structure that's very distinct from the one formed by residues 311 to 360. So we turned to get even higher resolution structural information, and that requires us to make sequence-specific assignments of the solid-state NMR data, and that's where we place these observed signals sequence-specifically into this protein sequence here. We record a series of two-dimensional and three-dimensional experiments that involve magnetization transfers similar to the ones shown here in the bottom left of the slide. And this is a pretty significant challenge because as I've been telling you about, these sequences are relatively degenerate, right? These are low complexity protein sequences. There are lots of similar amino acids in them. However, we have computational tools to tackle this problem. The only thing I wanted you to take away from this slide is that we have a computational algorithm shown here on the left that was developed by Rob Tico and co-workers, and that we've applied it successfully to several low complexity domains. So we're getting relatively good at making these kinds of assignments. And this is the result. So I just have some spectra here on the left that show you that we were able to assign many of these residues. And interestingly, all of these signals were assigned to a region between residues 365 to 400 of the C-terminal end of the low complexity domain, which is firmly outside the aggregation core that's previously been the focus of so much attention. We can use database predictions using the observed and assigned NMR chemical shifts to predict backbone torsion angles. And what we learned for this particular case is that there's regions of large negative phi torsion angle and large positive psi torsion angles, which are indicative of beta strands. So we know that there are at least 4 beta strands, or at least likely to be 4 beta strands within this low complexity fibril core. So if we can just bring it back to what we knew at the start of these experiments, was that there was a helix to fibril transition inside the aggregation core. Our contribution was to observe that in the presence of the full-length low complexity domain, not the full-length protein, but the full-length low complexity domain, that this second core prevents the aggregation core from forming presumably pathogenic protein fibrils. The fact that there's competing fibril cores in low complexity sequences is not unheard of. If we return to the fused in sarcoma example, shown here on the left, is the structure that we determined in Robtiko's group. But if you cut off the region that forms structures in the full-length low complexity domain, the C-terminal domain readily forms these beautiful looking protein fibrils shown here on the right. So this leads us to think about this in the context of the actual cell biology. We have this aggregation core that promotes the rigidification of stress granules and forms pathological aggregates, presumably accelerated by disease mutations. And this second core prevents the pathological association and perhaps allows some of these more functional activities to take place or to be propagated by TDP43 low complexity domain. All right, so now I'm going to go into depth about the most recent project from our lab, which is that relating to fibril seeding and the differences between that and liquid droplet aging for the TO1 protein. And this again is on the bioRxiv preprint server. If we can look back at the low complexity sequence, this is a much shorter low complexity sequence. It's 96 amino acids in length. It has slightly different biases of amino acids. We no longer have serine residues, but we do have a large population of proline residues. If we return to our histograms over here on the left, you can see that again, the TO1 protein is highly enriched in polar glycine amino acids and does have significantly more hydrophobic amino acid content than the fused in sarcoma case. So again, just wanted to point out that the TO1 protein does have N-terminal RNA binding domains, so it has a similar domain structure to the TDP43 protein, somewhat similar functions, but they're not the same. Here I'm representing the TO1 protein in the same orange squiggly lines. Like I said, the functions are not all the same, but they are involved in similar processes. And what really got us excited about the TO1 protein is that there's 7 disease mutations in this low complexity domain that influence its behavior. So work from Paul Taylor and Rosa Rademacher's lab identified these mutations in a set of clinical data. And they looked at 3 of these mutations in particular. And this is full-length protein that's been purified in the test tube. So there's some able to form droplets. You can look at these beautiful fluorescence microscope images on the left, but really what I want you to take away from this is the histograms on the right-hand side. And the top one shows that the recovery time in photobleaching experiments is increased for the disease mutants. And that means that the TO1 protein is less mobile within the liquid droplets. And the mobile fraction, or the amount of the protein that partitions into the liquid droplets, is going to increase. So the decreased mobile fraction means that there's more protein in the liquid droplets. So they also turned to some cell culture assays. You can induce stress in cell culture by adding things like arsenic. These punctiform, these are stress granules, which are similar to the liquid droplets that I was telling you about for TDP43. And the effect of these disease mutations is an increased persistence lifetime of the liquid droplets. The liquid droplets, or excuse me, the stress granules will persist for longer periods of time when you have these disease mutations present. And what was particularly interesting about this is if you look at the patient data, the disease mutations in TO1 are not actually associated with the buildup of inclusions of the TO1 protein. They actually are associated with the buildup of TDP43 inclusions. So these histological stains shown here on the bottom left are actually immunoreactive for TDP43, not TO1. And the data on the right-hand side is basically showing that TDP43 also loses mobility when you have the TO1 disease mutations present. So there's also other groups working on this problem, and they've been primarily focused on proline to hydrophobic mutations. So in addition to the clinical mutations I just discussed with you earlier, the idea that prolines could prevent pathological aggregation by disrupting secondary structure, and then converting those prolines into leucines could facilitate the formation of amyloid-like protein fibrils. This is just some of the data that they used to reinforce this hypothesis. For example, this serine 349 to proline mutation is actually the introduction of a proline, and it actually increases or retains mobility of the protein in liquid droplet type structures. And then these proline to leucine mutations here on the bottom actually increase the recovery time. So they're reducing molecular motion in the droplets. And I don't have time to tell you about all the data that went into their paper, but they came forth with this proposal that there are proline to hydrophobic mutations in TIA1 that extend beta strand structures and favor the formation of pathogenic amyloid-like protein fibrils, right? So the extension of a beta strand due to one of these mutations would then favor the formation of pathological aggregates. So a final piece of information that I find particularly interesting was a proposal put forth by Joseph Raymond and Eric Kandel from Columbia University, where they have proposed that the TIA1 aggregation could be both functional and pathogenic, right? So up here at the top is a diagram of the process I've basically just described to you, which is that you can get the formation of pathological aggregates, kills the cell, results in disease, but you could have limited fibril formation induced by environmental stress, perhaps by a seed structure that might exist endogenously in the cell, and then somehow that would be protective or could turn on or off different cellular pathways. So to investigate this, what we did in my lab is we produced the TIA1 low complexity domain by itself. It's the structure with the disease mutations are shown up here in the upper left. We were able to prepare seeds of the TIA1 protein. So we did this at near neutral pH, but very low ionic strength. And we were able to produce supersaturated solutions of the TIA1 protein. We mix those with the seeds. The seeds then template structural transition in the disordered monomers, and then we get the formation of fibrils. And I use the word limited here, not because we get limited fibril formation in the test tube, but the idea that these long fibrils that I'm going to show you are not necessarily the ones that would exist inside a living cell. So shown here is the result of our seeding assay on the negatively stained transmission electron micrograph. On the left-hand side shows a mixture of fibrils and amorphous aggregates. After our seeding procedure, if you look at the micrograph on the right-hand side, you can see that we get primarily long, thin, and somewhat bundled fibrils in our sample. So we put these into the NMR magnet. Again, these are solid state NMR spectra recorded under cross polarization conditions. So we're only looking at signals from the highly immobilized regions of the protein fibrils. We can immediately identify certain signals based on their amino acid chemical shift distributions. So we can see that there's a fair number of proline, alanine, glutamine, asparagine, tyrosine, serine, and threonine residues. I can't forget about glycine down here at the bottom. But what's also interesting is that there are a subset of signals that we observe in these 2D spectra. They're relatively weak, but when we go on to do the 3D spectra that I was describing to you for the assignment process, we don't see some of these signals. And in particular, I'll just draw your attention to the isoleucine residues, which are located at the N-terminal part of the TIA1 low complexity domain. So that suggests that these residues are immobilized enough to allow carbon-carbon or proton-carbon transfers, but they're not immobilized enough to give rise to signals in the 3D spectra. And you might be wondering whether or not we had enough signal to noise to observe these signals in the 3D spectra. So I just wanted to show you some planes from one of our 3D experiments shown here on the right. And these are relatively good signal to noise for these types of solid state NMR spectra. So we do believe that these signals are just not present due to molecular motion in the three-dimensional spectra. So we went on and again, went through the assignment process for these particular fibrils. I just wanted to remind you that we have a set of 2D and 3D experiments to do this, a computational algorithm that we have a lot of faith in. This is the result. So we identified a fibril core between residues 338 and 357 in the TIA1 low complexity domain. It's a 20 residue segment that is almost exclusively predicted to be in a beta strand conformation. There are two interesting aspects of this are that there's two ordered prolines. If you look at the C terminus of this 20 amino acid stretch, you can see that there's two well-ordered proline residues, and that most of the TIA1 disease mutations actually lie outside of the rigid fibril core. So to compare the seeded fibril preparation, which didn't go through a liquid droplet state, we decided to prepare liquid droplets and allow them to age, and then characterize their structures using solid state NMR. And we expected them to look very much the same. So this is some negatively stained transmission electron microscopy and some bright field microscope images of the wild type protein going through the aging process. And you can see, similar to the TDP43 case, we had the loss of liquid droplet structures going through increasing the amounts of molecular order, resulting in these pathogenic or these fibril-like structures in the upper right, shown here. We did this for a P362L mutant. So this is testing the proline to leucine hypothesis. And we see a very similar loss of liquid droplet structure and the rise of fibrillar structure based on the electron micrographs. We used fluorescence to characterize the bulk phase behavior of these proteins. Shown here on the left are the thioflavin T assay. And what we see after 1.5 hours are these blue curves here. The dashed line is the mutant, the solid line is the wild type. We see an increase in thioflavin T fluorescence. What was interesting is that 4 hours, we lose thioflavin T signal before gaining it back again as the fibrils form over the span of about a week. Tryptophan fluorescence, again, this is measuring the accessibility of tryptophan residues. To solvent, we have 5 tryptophans that are relatively well distributed throughout the TO1 low complexity domain. And you can see we see a significant reduction in intensity as well as a shift to lower wavelengths. Interestingly, there is no, substantial amount of quenching of the signal at the 4 hours. And the thioflavin T and the tryptophan fluorescence data suggests that this intermediate state, this amorphous aggregate state, is not cross beta in nature. So something happens, it's the transition of fibrils through a non-cross beta state on the way to the cross beta type fibrils shown here on the right. So we put these samples into the NMR magnet just to show you the electron micrographs of the fibrils that we put into the spectrometer. These spectra are shown here. They're very similar to one another, but if you're not used to looking at these things, we can do a different spectrum where we subtract the spectrum on the right from the spectrum on the left, and it's much easier to see similarities and differences between these two spectra. So if you look at the spectrum here on the right, it's essentially nothing but noise. There are a few signals in here suggesting that there's differences with threonines, valines, prolines, and perhaps leucine residues, which might be expected based on the mutation. But in general, the take-home message from this different spectrum is that the spectra on the right, or excuse me, on the left, are very similar to one another. So both the wild type and the mutant droplets age into essentially the same fibrillar structure. We can compare this again to our seeded preparation. So the spectrum here on the right in the center is the seeded fibril preparation, and the one on the left is the wild type aged droplet spectra. So the electron micrographs are shown here. So these fibrils do look somewhat different in the electron micrographs, but it's very difficult to tell if there are actually significant differences. Most interesting to us, immediately looking at these spectra, if I can just draw your attention to these 2 black squares here, is the loss of ordered threonine signals in the wild type and aged liquid droplets. There are other differences, but these are probably the easiest to see at first glance. And the other defining feature of the aged droplet spectra is this broad NMR line shapes. Okay, so the NMR line shapes could be due to molecular motion, or they could be due to the presence of several molecular conformations for each amino acid in the T1 low complexity domain. So we did some relaxation measurements. So we measured C13 and N15 T2 measurements. The results of these tell us that each NMR line shape in the spectrum should be about 0.5 part per million wide, which immediately tells us that the broad spectrum here on the left is due to conformational heterogeneity, not a single conformation undergoing dynamics to give it broad lines. We can repeat our different spectra approach where we've subtracted the spectrum in the middle here from the spectrum on the left. And the interesting part here is that the signals, the sharp signals that report on the seeded core of the T1 fibrils is the only thing left when you do the different spectra. And what that means is that the aged droplets exhibit or contain very little structure that's consistent with the seeded fibril preparation. So although there are heterogeneous conformations in the aged droplets, they are not the same in general as the seeded low complexity domain fibrils of TO1. Okay. All right, so that led us to put forth the following model based on our observations, which is somewhat similar to the Eric Kandel model, where we have disordered protein monomers. In times of stress in cells, you form condensed liquid droplets or stress granules. If these are allowed to age, perhaps due to the presence of disease mutations, you get the formation of heterogeneous fibrils. These are, either collections of fibrils of different structures or fibrils that contain molecules in different conformations, which is perhaps unlikely. And that these heterogeneous fibrils are the types of structures that would permit or favor the aggregation of other proteins like TDP-43. So this could explain the disease pathway. And then if you look down here at the bottom, the disordered monomers could undergo a seeding process, maybe through templated growth through an endogenous seed that was proposed previously. These are allowed to elongate, maybe under limited lengths, to form homogeneous fibrils that could facilitate functional protein-protein interactions related to functional activity. Okay, so that about does it for my talk. I want to just reiterate what I told you today, which is that the TDP-43 low complexity domain contains multiple fibril cores. We have suggestive evidence that there's a second core that prevents aggregation or pathological association of the aggregation core. And then we looked at the TO1 low complexity domain, where there's a seeded fibril core that seems to be distinctly different than the heterogeneous fibril formation that you get out of aging of liquid droplet structures. So I have to thank my team who did all this hard work. This is our most recent lab photo. We're glad to be back in person. Blake Fonda, Khaled Jami, Yuki Whitmer, Truk Lee, Natalie Boulos, and Rochelle Stoll were the ones that did the specific work that I told you about today. I have to thank the NIH and the University of California of Davis for funding. We are looking for postdocs, so if you'd like to check out our website or send me an email, I would really like to talk to you, and thank you for your attention. I'll take any questions.
Thank you, Dylan. That was really exciting, and you're already having 2 questions in your Q&A. The first one is from Robert Tico, and is there a significant difference in amino acid composition between the traditional aggregation core TDP-43 and the alternative core identified by US solid-state NMR measurements?
So I cut out in the middle there, but the question is the sequence similarity between traditional amyloids for the TDP-43 second core?
Yeah, and the alternative core identified by US solid-state NMR measurements.
Yeah, so that sequence is much more enriched in glutamine amino acids. The methionines and alanines that are so present in residues 311 to 360 are not present in the secondary core. So it does seem to be less hydrophobic than the aggregation core.
The next question is from anonymous attendee. Great talk. How did you extract the torsion angles from new solid-state NMR data? Did you record NCCN-based experiments? Can you comment on that? Thank you very much.
Yeah, right. So it would be a great idea to measure those specifically. But what we did is we took the assigned NMR chemical shifts and we put them through the TELOS-N database prediction program. So these are based on existing structures, not experimental measurements other than the chemical shifts that we observed.
Thank you. And the next one is from Marcus Zweckstetter. And the question is, what is the contribution of the N-terminal disordered region of TDP-43 to misfolding aggregation of full-length TDP-43?
So I assume that they're referring to the very N-terminal domain rather than the N-terminal domain of the low complexity region. And there is evidence that those interact Folks like Nick Fauzi have looked at that. So there is a role. What we observe, or we're starting to observe, is that depending on what we put on the N terminus of the low complexity domain, that does seem to alter what happens, but we're still trying to sort that out. And there is a solid state NMR study of full length TDP43 that gives a somewhat different signature. So we were using the alanine residues that we observed as sort of a metric to compare different TDP43 structures, and it does seem to be somewhat different. I don't think that the balance between the pathogenic aggregation core and the sort of secondary core that we identified is not relevant in the full-length protein. I think that it's just a little bit more complicated that, and this is just one piece in terms of trying to understand how these interactions play out in the full-length protein.
Thank you. You have another anonymous attendee asking, saying thank you for the great talk. How the low complexity domains are related to intrinsically disordered ones.
Right, so not every low complexity domain would be intrinsically disordered, I suppose. And the low complexity domains I told you about are a subset of low complexity domains. So like the nuclear pore proteins have very different properties. They're highly hydrophobic sequences. So I don't know if I can comment in general about the differences or the similarities between low complexity sequences and intrinsically disordered regions. They do tend to be disordered, at least the ones that we studied, but there are low complexity regions that do adopt well-defined secondary structures. So I know that's not much of an answer, but that's all I have.
The next question is from Parvez Alam. Great talk. C-terminal domain of TDP43 is prone to aggregation in solution. How do you control that?
Carefully. So when we purify it we produce all our material using bacterial expression systems. We use guanidinium and urea denaturants to keep the protein from self-associating, and we can remove that either using buffer exchange techniques or dialysis. We are able to handle the protein at 20 micromolar. So Khaled Jami and my group has been able to do things like cleave off the his tag using 20 micromolar protein concentrations. And if you handle it well and you have it well purified, you can actually do quite a bit with it in terms of reconcentrating the protein and things like that. But it's a challenging process for sure.
The next question is from Rams. Excellent talk, Dylan. Did you try lowering the temperature to reduce the mobility of residues like isoleucine to enhance the signal to noise ratio in 3D experiments?
Yeah, so we did do that. This is what, I don't know if I'm still, yeah, I'm still showing the screen, but the spectrum here on the right, spectra on the right, are actually the low temperature spectra. And we expected the 3D residues to show up in these, and they didn't. So we're comparing plus 10 Celsius with minus 20 Celsius. That's what the temperatures that we had access to. Proline residues are another reporter. If you look here in the bottom right, these are actually TDOR type experiments, which are better used to observe rigid prolines, and we don't see those ordered either. So our interpretation is that they're just in heterogeneous conformations, so lowering the temperature doesn't solidify them into something that is similar to the seeded fibril conformation. So I hope that answers your question.
Yeah, thank you. I think for the sake of time, we are just running on time. We will move to our next speaker, Dr. Henry Paulson. Dr. Paulson, do you mind if you share your screen? Sure. And while we are switching speakers, it's my great pleasure to introduce Dr. Henry Paulson, who is the Lucille Groff Professor in Neurology for Alzheimer's disease and related disorders in the Department of Neurology at the University of Michigan. Dr. Paulson joined the University of Michigan faculty in 2007 And he currently directs the Michigan Alzheimer's Disease Center, MADC. He also co-directs the University of Michigan Protein Folding Disease Initiative, which has been really great in uniting all the scientists within UM to understand the protein unfolding and its implications for the disease. And he also co-directs the Michigan Neuroscience Institute. It's also at U of M. Dr. Paulson has multiple awards. He's an Allison Medical Foundation New Scholar in Aging. He's a semi-finalist for the Keck Foundation Young Scholars in the Medical Research, and a recipient of the Paul Beeson Physician Faculty Scholar in Aging Award from the American Federation for Aging Research. He's also a member of the National Academy of Medicine. Dr. Paulson's research and clinical interest concern the causes and treatments of age-related neurodegeneration diseases. He pioneered a lot of developments in that disease, in particular in the polyglutamine diseases, Alzheimer's disease, and frontotemporal dementia. He has been also my mentor, and while I'm here in the UFM, we're in the same department. And thank you so much for joining our ZOOMinar series, Hank. And it's your turn. I don't want to steal any more of your time.
Thank you, Magda. I appreciate that very kind introduction. And can you see my slides?
Yes, your slides are beautiful.
Okay, thank you.
And I don't know how you got the picture of our building.
I took that picture. My office is right here. Magda's office is right here. Sammy Barmada's office is right here. So there's quite a few people who work on neurodegenerative diseases here. Peter Todd's is right around here. Anyway, I just want to say, Rams and Magda, this is a great ZOOMinar series you've had. I think you know that I have a standing meeting on Mondays that makes me miss almost every single one of these, and I apologize for that. And hearing Dylan's outstanding talk makes me realize how much I've missed out on some fantastic science. As I go through here today, I want you to think about polyglutamine diseases in the context of what you heard from Dylan, because I think neighboring structures and how they affect sort of low complexity domains like a polyglutamine domain are something we need to be thinking about more. I want to just simply acknowledge that the work that I talk about is not done by me, but it's been done by people who've been in my lab or in my lab. I'm just highlighting a number of them right here. For the sake of time, I won't go through them individually, but they've all had tremendous contributions. I'm not including the faculty like Magda or Sammy or Peter, who I've worked with and who've influenced me greatly. You know these folks. You just heard Dylan talk about TDP43 and TIA1, clearly involved in ALS. All of these diseases have protein accumulation. I'm going to talk today about polyglutamine disorders. Woody Guthrie, of course, had Huntington's disease, which is a purely genetic autosomal dominant disease due to a CAG expansion that encodes glutamine. There are at least 9 different glutamine diseases. My lab works broadly on mechanisms of neurodegenerative disease, and our emphasis right now is on proteins and pathways that are linked to ubiquitin. If you think about ubiquitin, the first thing you think about is the ubiquitin proteasome system, but the reality is ubiquitin is engaged in so many different pathways in the cell. And I would submit, although we don't know enough about this yet, that in neurodegenerative diseases of various sorts, ubiquitin homeostasis is impaired in ways that affect far more in the cells, neurons, and other cells of the brain than the ubiquitin proteasome system. Here I'm showing you a picture from I Heart Huckabees, the movie. Here's Dustin Hoffman. He's holding up a blanket. He's talking about universal interconnectivity. Something happens over here, sort of like the butterfly flying, influences something that happens over here. I think the reality is with ubiquitin pathways, when there is perturbations of ubiquitin homeostasis, it's not simply affecting ubiquitin proteasome system, but in numerous pathways in the nucleus and outside of the nucleus. And our emphasis is on 2 major disease proteins. One is ubiquitin 2. I'm not going to talk about that today. I'm sure Magda has spoken about that, and we've had the privilege of collaborating with Magda and others to study the ubiquitin proteins, which are ubiquitin shuttle proteins that bind ubiquitin and have a ubiquitin-like domain. And today I'll just talk about ataxin 3, which is a polyglutamine disease protein that causes spinocerebellar ataxia type 3. It is a deubiquitinase, and we'll talk about that more later. There are many different repeat expansion diseases. This is a schematic of a gene with many different locations for different repeat expansions that cause various neurological diseases. The ones that we're going to talk about today are the polyglutamine diseases. These are due to a CAG repeat expansion that's in a protein coding region in frame to encode glutamine in the various disease proteins, such as spinocerebellar ataxia type 1, 2, 3, 6, 7, 17, Huntington's disease, spinobulbar muscular atrophy, which is a motor neuron disease. These are clinically distinct disorders. No one would confuse SBMA, which is a motor neuron disease, with SCA3, which affects the cerebellum and the brainstem and causes ataxia, and would not confuse that with Huntington's disease, which causes more of a degeneration of the basal ganglia and the cortex. They had the same underlying mutation, a CAG repeat expansion that encodes glutamine in various different disease proteins, and yet clinically they are distinct disorders. Even the SCAs, the dominantly inherited ataxias, can be distinguished to some extent from one another. SCA3 is the one I'll be talking about today. And again, they encode in the normal proteins a stretch of glutamine. And in the disease situation with the CAG expansion, they encode an enlarged stretch of glutamine. I'm not addressing at all the potential contribution of RAN translation repeat associated non-methionine translation, which can occur in CG-rich expansions where there can be, if you will, different frame, non-methionine driven expressions. In this case, would make polyserine, polyleucine, but in fact, I'll be focusing on the polyglutamine. My bias is that you don't need necessarily to invoke RAN translation peptide products in order to understand the disease process. Is there RAN translation Huntington's disease? Clearly, and I think there probably will be data to show that it's a contributor to various CAG repeat diseases. But we'll focus here more on the disease protein itself. The disease protein in SCA3 is ataxin 3, and the expansion that occurs goes from a normal repeat of 12 to 40 up to between 60 and 80. The longer the repeat, the more severe the disease. Most people have disease, have ataxia that occurs midlife. People with a longer repeat have an earlier onset disease that has a slightly different flavor. It has more dystonia than ataxia. And people with a smaller repeat in the 60 range have a later onset disease that has more motor weakness than dystonia. So if you will, there is a dose dependence to this, a length dependence to this. The longer the repeat, the more severe the disease occurring earlier in disease. And people who have an intermediate size repeat don't have disease itself. They have a syndrome called restless leg syndrome, which may be unrelated to what we see here. Now, in all of the polyglutamine disorders, we see evidence of aggregation of the disease proteins. I'm just showing you examples here from human disease tissue of a pontine neuron, showing an ataxin 3 inclusion that's also ubiquitin positive, or from SCA3 knock-in mice, in which this red is indicating aggregation of the ataxin 3 protein in the hippocampus, or here in cell models where we've expressed just a fragment, an expanded polyglutamine fragment behind GFP that forms inclusions in every cell that you see in the dish. This can be seen in many model systems. It's seen in every single polyglutamine disorder. And it's led to a question that's been in the field for a long, long time. What's the relevance of polyglutamine aggregation to neurodegeneration? And there are many different camps that have different views about this, for everything from, well, they're purely protective, that is, the aggregates occur and that you have protection, to they're an epiphenomenon of disease. Yes, aggregation occurs, but they're not at all relevant to the disease. To another view, which is that aggregation occurs, it has bad properties in some ways. It can mean many different kinds of bad properties and is a contributor to toxicity. I tend to be more on that last category, which is it's not a good thing to have aggregation. And if the only thing it's doing is actually causing a constant burden of abnormally folded protein, that in itself is going to have deleterious consequences for cells. I'm not going to go through this in detail, but over a long period of time, we've looked at a lot of different ways that aggregation or misbehavior of the protein could contribute to disease. employing model systems in collaboration with Nancy Bonini in flies to mouse models, looking at whether aggregation affects the proteasome. It does indeed. Whether heat shock proteins are involved, they clearly can be involved. They can be sequestered in aggregates. And if you overexpress heat shock proteins, you can see that you can mitigate some of the disease features. We can see that proteins like Krebs binding protein, which is a critical gene regulatory transcription cofactor in the nucleus, becomes sequestered when you have expanded polyglutamine around. We see that in multiple model systems. And finally, this has been a work that's come back many times again, is that the specific aggregation that occurs is specified by the protein composition and neighboring domains, not unlike what we heard from Dylan with TDP43, can really influence the behavior of the aggregation core of the protein. I'm going to really here, however, focus on an early study we had in 2002 in PNAS, which suggested that disease protein behavior was far more complex than simply aggregation. So we went into the study generating a series of constructs in which we expressed a fluorescent tag behind either huntington fragment or ataxin 1. Ataxin 1 is not ataxin 3, but I'm simply showing in this paper that we looked at ataxin 1, huntington, as well as ataxin 3, which I'm not showing you here. And then we did fluorescence recovery after photobleaching or fluorescence loss in photobleaching and looked at the behavior of these particular aggregates. And we really wanted to see if they actually were truly fixed, if you will, aggregated protein in the nucleus. And so here is an example of a nuclear inclusion of a huntington fragment, which got zapped halfway through. And then we followed over time. And you can see that really this is a fixed structure. You're not seeing bleed of the fluorescence from one half of the inclusion to the other. Likewise, when a cytoplasmic inclusion of huntington was bleached in the same way, you don't see a significant amount of recovery. What surprised us most in this paper was that there was a lot of dynamic behavior of inclusions, and we didn't understand at the time, and I think now we have much better insight into that. So here, for example, we bleached an entire inclusion of an ataxin 1, if you will, inclusion of protein in the nucleus of cells, and followed over time. And you can see there's a very rapid recovery. So there clearly is mobility of the proteins that are now going into these inclusions. If you zapped half of this, you saw tremendous mobility within it. These were not aggregates. These were actually, if you will, biological condensates with mobility, liquid-like structures in the cell. We wrote in the abstract, there's markedly divergent mobility states for an expanded polyglutamine protein ataxin 3, and unexpected heterogeneity in the dynamics of inclusions formed by different disease proteins. At the time, we certainly did not have a physicochemical view of this. We did not think hard enough about this, but I think everyone would agree that what we're looking at really is the behavior of proteins. Now we can see them in a different set of lenses that are engaging in biological condensate formation, engaging in liquid-like structures. And so here is not a paper from us, but this is a picture from Pescott et al. Showing the behavior of a fragment of huntingtin that goes through a liquid-like structure. It can form solid-like fibrillar structures as well. And the same is surely occurring with ataxin 3, that either the normal protein or expanded polyglutamine containing ataxin 3 is engaging in different structures and probably driven more towards a solid-like structure when you have the aggregation. An interesting feature of ataxin 3 is that it's a fantastic binder of ubiquitin. It loves to bind ubiquitin. Obviously, it's a deubiquitinase and it cleaves ubiquitin chains. And almost certainly the ubiquitin environment in cells is influencing the behavior of ataxin 3 and probably relevant to its function normally and its dysfunction in disease states. We know that this drives many cellular processes. The relevance of biological condensates to, and liquid-like behavior of polyglutamine proteins normally and in the pathogenic forms is a work in progress. A lot more needs to be done here. This is definitely true for ataxin 3, but I would say huntingtin has been studied more than anyone, ataxin 2 probably the number 2, and we still don't know nearly enough about how these glutamine domains normally influence the behavior of these proteins and how when expanded they drive disease features. And so a very simplistic view for this audience is that the idea of the amyloid aggregation, if you think about, this is an old figure from a paper from Chris Ross and Natalie Poirier. And if you think about this, is not unlike beta amyloid, which is a fragment of the APP protein that's taken out of the cell, out of the protein. Pure A beta 1 to 42 is very prone to form amyloid-like structures. There's nothing that actually is inhibiting that. And you see this occurring quite well. In the case of the polyglutamine proteins, that polyglutamine domain and the threshold for aggregation is for most of these proteins about 40. Beyond that, clearly aggregation prone, less than that, much less aggregation prone. But in this case, it is flanked by various domains that differ greatly from one polyglutamine protein to the next. And these are just a schematic of two different proteins. And you can imagine that these other domains not only, if you will, inhibit the formation of oligomers or aggregates from the polyglutamine domain, but actually in themselves may influence the behavior, not unlike the the neighboring domain that Dylan described in TDP43 that is influencing the aggregate core in that protein. We think that the polyglutamine domain is intrinsically disordered. I don't know if you call it low complexity, but certainly can adopt different conformers. And that is relevant, I'm sure, to its normal function. I like to think of ataxin 3 and other polyglutamine proteins as having a glutamine domain in part because it allows for conformational flexibility. Polyglutamine proteins, polyglutamine disease proteins, are typically not hardcore structural enzymes. They are proteins that exist in multiple different macromolecular complexes, and in some cases, many different complexes, and being able to be flexible enough to engage in different complexes may require some degree of conformational flexibility in the polyglutamine domain. The normal stretch probably allows for that dynamic behavior and interchange between conformers that help to maintain the function of the protein. When you have the expansion of a polyglutamine domain, there's good evidence. We don't have great structural data on polyglutamine, but there's I think enough biophysical evidence to support and biochemical evidence to support the idea that an expanded polyglutamine domain can engage in intramolecular structure that is an energy state that is difficult to get out of, and that when it's, if you will, sticking out of the protein, it's probably prone to form more ordered structures like this. And I'm also showing you here the glutamine domain that has been cleaved out of the protein. And we know this occurs in huntingtin and occurs in some other disease proteins, is that in the disease state, under the stress of disease, there is proteolysis of the disease protein, and you're more likely to have a polyglutamine domain that is untethered by surrounding domains. So when you have the expansion, there's a loss of probably the normal dynamic interchange. That's going to change normal function. You have an increased propensity to engage in aggregates, and that aggregation is enhanced by proteolysis. These are the different disease proteins. They are completely unrelated at a primary amino acid structure, and they don't seem to have much similarity at the secondary or tertiary structure. Each of them has a different part of the protein in which the glutamine domain is contained. I initially started working in huntingtin as a postdoc. It's just a massive protein. Ataxin 3 was cloned, and I switched over to ataxin 3. We could make recombinant protein pretty easily and study it. Even though it's a lot smaller, it's still a very complex protein. When you think about ataxin 3, it has a very highly conserved amino terminal globular domain. We do know the structure of this. And it's the catalytic domain of the enzyme. It's a deubiquitinase. The tail of the protein is more flexible. It has 3 different ubiquitin interacting motifs that are flanking the polyglutamine domain. This protein loves to bind polyubiquitin chains. It has at least 6 different ubiquitin binding domains, not simply the UAMs, but other ubiquitin binding domains in the ataxin 3 domain. The josefin domain, and it can actually be directly conjugated to monoubiquitin that seems to increase greatly its DUB activity. What we've learned over the years, and this is work from our group and others, is that this DUB is fantastic at cleaving very long polyubiquitin chains, regardless of their specific chain structure. They're poor at cleaving short ubiquitin chains. They do a terrible job of cleaving a tetraubiquitin that's K48 linked. They can do a fairly good job of a hexaubiquitin chain, particularly if it's got mixed linkage chains, and it seems to do a better job with K63 linkages than K48 linkages. Expanded ataxin 3 in a test tube still does a pretty good job of binding polyubiquitin, and expanded ataxin 3 in a test tube, again, still does a pretty good job of cleaving polyubiquitin. We don't know nearly enough about this protein in the actual physiological state of a cell. And that needs to be addressed now, and we're trying to get at that ourselves. Our current view, based on data that interacts with numerous ubiquitin ligases is that it actually is a ubiquitin chain editing enzyme that partners with ubiquitin ligases to regulate ubiquitin chain composition. And even though we know expanded ataxin-3 still interacts with parkin or still interacts with CHIP, how that influences the quality control DUB activity in vivo is still a work in progress. So what are the potential contributors to toxicity? Clearly, the concentration of the protein in the cell, we believe is important in terms of driving towards oligomer state and fibril state. We think that this can alter the normal function. We think it's very likely that expanded polyglutamine leads to neofunctions, that is, new interactions. Some of them might be deleterious, some might be beneficial, we don't know. When you have a constant burden of abnormal protein in the cell, this is a stress on the quality control pathways, including particularly ubiquitin link pathways. There's evidence that ataxin-3 forms aggregates that are stuck in the processes of neurons. That can't be a good thing. It's a physical perturbation of the cellular milieu. We don't think that loss of ataxin-3 function is a major driver of disease based on genetic data and cellular data and knockout mice data. We think rather it's more of a toxic gain of function. And again, the relationship between that above oligomer fibrillar inclusions to the biological condensate normal properties of this protein is something that needs to be addressed more thoroughly. We've used many different model systems to look at how ataxin-3 behaves normally and misbehaves. And like you all, I'm sure, use more than one model system to look at problems, in part because no model is perfect. As George Bach says, all models are wrong, some are useful. And I think the combination of models can be particularly useful in times. I'm just going to talk a little bit about some invertebrate data and mouse models that we've used. Here is a yak transgenic that overexpresses normal ataxin-3 gene, the full human gene. And this is staining for ataxin-3 shown at different ages in the deep cerebellar nucleus. And you're not seeing much staining of ataxin-3. When you have the yak transgenic as a function of time, you now begin to see the protein concentrating in the nucleus. It's normally more present in the cytoplasm. And if you have a homozygous mouse, this occurs even to a greater extent. We've used that mouse and many other mouse models to begin to address that question of whether, what is the relationship between aggregation that we see in these mice and gene expression changes as a potential proxy of toxicity. So we've taken mice that express expanded ataxin-3 or normal ataxin-3, or we've taken knock-in mice that have a low level of aggregate phenotype and mice that have a higher level of aggregate phenotype or a moderate level. And we've taken those mice at an early phase of disease state, and we've looked to see if there are changes in gene expression. And so just looking at two mouse genotypes that have high level of aggregation and one that has a modest level of aggregation, and looking at gene expression changes using RNA-seq at a fairly early age before we see lots of pathology. I'm just going to highlight here genes that are downregulated and upregulated that are highly aggregation prone, boxed in blue, or those that are moderately aggregation prone that are boxed in orange. And I think you can appreciate there's shared gene expression changes. When we actually map this out and say, are the gene expression changes more increased when you have more aggregation? They absolutely are. You see the most gene expression changes up and down regulated when you have the highly aggregation prone mice. So this says, yes, indeed, there is a correlation between aggregation and gene expression changes. We can't make any causal links. But the intriguing thing for us when we did this, the most surprising thing by far, was that we expected, because we have a neuronocentric view, is that we would see lots of gene expression changes in neurons. But in fact, what we saw were gene expression changes in oligodendrocytes far and away the most. We didn't expect that, but now data are showing increasingly that oligodendrocytes and white matter tracts in disease are affected very early. We did not know that, but the mouse models have helped to influence our views of the human disease data, including imaging in humans. So Haley McLaughlin, who was a postdoc and now is an assistant professor in our department, has really gone with this. And these are really Haley's beautiful data, looking longitudinally over time at gene expression changes. And sure enough, when she looks for the kinds of modules of related gene expression that are changed over time, the turquoise modules showed the greatest changes as a function of age. And where are they predominantly expressed? really there in oligodendrocytes, either oligodendrocyte precursor cells or myelinating mature oligodendrocytes. So again, the data are pointing to an early effect in these diseases, in this disease, and now we know in other polyglutamine diseases of oligodendrocytes. And Haley and Kristen in her lab have gone further to actually cross our mice to a myelinating oligodendrocyte reporter mouse line. So you can see a mature oligodendrocyte in these mice because they are fluorescing for green fluorescent protein, and wild type mice show beautiful early maturation of oligodendrocytes in the pons and the deep cerebellar nuclei and in the corpus callosum. But if you look at the polyglutamine SCA3 mouse model, you see marked delay in the maturation of oligodendrocytes, particularly in those vulnerable regions of the pons and of the deep cerebellar nucleus. There's very little evidence that the corpus callosum is involved in human disease, and likewise, we see much less involvement in the corpus callosum in these mice. So this is a work in progress, but the reality is we now can see that oligodendrocytes are one of the earliest affected cell types in this particular polyglutamine disease, and data are accumulating in other polyglutamine disorders that we need to be looking more at non-neuronal cells, even than neurons. So the data today really have supported the idea that there's a toxic gain of function, whether it's aggregation or whether it's aggregation plus other things or whether it's other things, and that we need to be thinking about that with respect to potential therapeutics. And so we've worked very hard over the last decade at looking at therapeutic targets in disease, and I don't have time to talk about that in detail, but among the approaches have been nucleotide-based gene silencing, genetic and compound screens to find factors that can reduce the disease protein. We think if we could reduce levels of the disease protein, that would be a positive, and possibly to find things that block toxic oligomers. Today, I'm really going to talk in any detail about studies in nucleotide-based gene silencing. We've used two basic approaches. One was co-opting the RNA interference machinery using small interfering RNAs or microRNA-like approaches and viral delivery into mouse models. And that's the work that Carmel Costa mostly drove. This has been published and very successful at silencing ataxin-3, but the virus, when delivered to the confined region of the brain, did not cure the phenotype in these mice. More recently, we have focused, and this is the work again of Haley and Lauren Moore, who's now moved on to be the Chief Scientific Officer at National Ataxia Foundation, to use antisense oligonucleotides to directly target the ataxin-3 gene. And using ASOs, and I think you've heard about this before, so I'm not going to go through this in great detail. We've identified many different ASOs that can target the ataxin-3 gene. Here we looked at 5 in mouse embryonic, human disease SCA3 fibroblasts that expressed normal and expanded ataxin-3, and all were very good at decreasing levels of both the disease protein and the normal version of the protein. We moved into mice where we injected the ASO in a single ventricle and then looked at the distribution of the ASO. You can see the ASO in green here went throughout the brain. And here we're looking at the expression of ataxin-3 in a wild-type mouse, or in the YAC transgenic mice, you see lots of ataxin-3. And this is the ataxin-3 mouse that got the control scrambled ASO. And here's the ataxin-3 mouse that got the targeting ASO against anti-ataxin-3 and marked suppression of ataxin-3. We showed that this was eliminating both the monomeric ataxin-3 as well as the aggregated ataxin-3, and that the ASOs worked for a fairly long time in these mice. You know, a couple months after you've injected it, you start to see accumulation of the protein again. So we realized if we're going to really see if ASOs work well, we need to be looking at multiple deliveries and then following the mice over time. So this led us to actually do a phenotype assessment where we injected at early, and then we followed them, and then we injected once again, and then did our final assessment 29 weeks. We did a behavioral assessment here just before the mice were harvested. And this is just showing you that there was a behavioral correction in these mice. If you look at this traversing of a balance beam, the YAC transgenic mice get slow over time. They're not very good at the balance beam, and wild-type mice do very well. These are the YAC transgenic mice that got the ASO, and they're basically largely corrected of the phenotype. So this was beautiful. And in fact, we showed that there was correction of the gene expression change that I talked to you earlier about. I'm not going to go through some detail, but this is, again, an oligodendrocyte expressed gene that is corrected when we add the ASO back into the mice. So the use of ASOs and the SCAs has been a real success story. We have basically cured the disease in SCA3 mice and SCA1 mice and SCA2 mice and SCA7 mice have all been cured with ASOs that target the genes themselves. And now we have a phase 1 study in humans of our SCA3 ASOs that Biogen is doing. We're delighted to see that happening. But I think you know that there are major barriers to therapy, and there's been failures in the huntington ASO, and I've heard that a C9 ASO trial has not gone so well. And there's a lot of reasons why we need to be recognizing that this may or may not work when we actually bring it to humans. We need to know more about disease progression, and we have a large study right now to understand disease progression in SCA3 and SCA1. We need a better knowledge of the biomarkers that we can track over time, and we're doing that, looking at for magnetic resonance spectroscopy changes that occur over time in a large multi-center clinical trial, and actually showing that we can target the disease protein. And now there are good assays available to look at that in CSF and hopefully soon in blood. We need more work to show that we can engage the target in humans, and that's a work in progress as well. And I'd say even when we have all those things, we would be stupid to put all of our eggs into the gene silencing basket. I mean, yeah, it might work. It's a compelling target, but we need to recognize that we need to be thinking beyond that. And although I don't have time today to talk about it, Carmel Costa has really been leading the charge, and she is also an assistant professor at the University of Michigan, now working independently, looking for both genes and compounds that can reduce levels of ataxin 3. And while we can't go through that in any detail, she's developed beautifully parallel and complementary cell-based assays and in vitro slice recording assays, and the mouse themselves, to go and look at compounds or genes to identify those that might actually be regulators of ataxin 3. And so in this particular screen, she looked at roughly 3,000 existing FDA-approved compounds, or compounds including many that are FDA-approved. And out of this, she found 3 compounds, including an existing antipsychotic known as aripiprazole, that did a great job of reducing ataxin 3. And when she injected these into the mice, in a very short preclinical study in the yak transgenic mice, there was a marked reduction of aggregated ataxin 3 protein. So in summary, what would I have to say is that, I think it's the principle of parsimony probably does not apply to any chronic neurodegenerative disease. And so if I say to you aggregation is the end all, I'm wrong. There are many different things that occur when you have a chronic disease state, and things are occurring over time. And I would anticipate that it's not simply the expanded polyglutamine protein itself that's misbehaving, but other things that occur as a result of that, or indirectly or directly. We do think there are more than one contributor to pathogenesis in the polyglutamine disorders. And the key to therapy will be really identifying those proximal steps or nodes in the cascades. I think we need to know a heck of a lot more about the normal properties of these proteins, including how the formation of biological condensates by these proteins actually may be critical to their normal function and may be critical to their dysfunction disease states. That we think that reducing levels of toxic gene products is a compelling, but not the only route to disease-modifying therapy. We need to know more about biomarkers, and I would say that, this is only one of many repeat expansion diseases. There are at least 6 new repeat expansion diseases in the last 3 or 4 years. There's a lot we don't know, and I would say of those of you who are protein biochemists out there, there's a lot that you could contribute to the field. I just wanted to stop by thanking the people who've done the work, and I'm highlighting in yellow faculty at the University of Michigan who we've worked with and had the pleasure to work with, faculty who I've worked with for years, Bev Davidson at Penn and Nancy Bonini at Penn, both of whom have been tremendous collaborators. In blue here are my co-investigators in this large multi-center clinical trial study. I showed you earlier some of the earlier people in the laboratory, and this is already out of date because there are 2 new people here that are not shown, but these are my current lab and some of the funding. And again, she mentioned the protein folding disease and the Michigan Neuroscience Initiative. So I will stop here and stop sharing my screen.
Thank you, Hank, for the great talk. And I just want to remind the audience to please post your questions in the Q&A or chat. And I will start with my questions first. In regard to ASO, do you think that it looks like there should be one therapy won't be sufficient to kill, like to deal with the disease? Like in the case of ASOs, it's having a very good reaction in the mice, as far as I understand, but it's in humans, there's simply still work in progress. But do you think like the double therapies, like targeting at the same time the aggregation, protein aggregation, for example, and using ASOs will be something which we need to focus? And why people are not doing it? I kind of
well, I think yes. I think the answer, Magda, is yes. We didn't, if you will, cure HIV disease until there was multiple therapies, and we know that anti-amyloid in Alzheimer's is not going to be sufficient. I think that Alzheimer's is going to be a multi-therapy disease when we finally have really good disease modification. I think that in a monogenic disorder, the idea of starting by trying to reduce levels of the implicated disease gene, as long as it's not an essential gene, is a good way to start. Now in Huntington's disease, obviously it's an essential gene, so it's a really difficult task to silence that culprit at the same time that you don't cause toxicity from that. That may not apply to ataxin 3, but I think you're right. I don't think we're going to have cures from single monotherapy here. So the idea that other approaches will have to be brought in seem relevant, but I realize as a clinician that until we have better ways to show an effect of a therapy in humans with biomarkers and better quantitative scales, we're not going to actually know the answer to that. I mean, we simply, there has to be parallel preclinical studies that look for multiple therapy efficacy at the same time that we develop better biomarkers, better quantitative understanding of the disease, so that when we have the dual therapy ready to apply, we can do it well. And one thing I will say is there are potential symptomatic therapies that modulate ion channels that also could have disease modifying effects. So the way of combining like a ion channel modulator with gene modification at the same time is one very compelling strategy.
Yeah, thank you. And in regards to oligodendrocytes, again, down the line of the multiplex therapy, you were like, what are the best avenues to pursue? Obviously, it looks like the disease first starts in there. So
one of the first questions was, do ASOs hit oligodendrocytes? And the answer is yes. Haley and Lauren were able to show that one, there is evidence of pathology. By the way, both my lab and Haley's lab are looking for postdoctoral associates, as is Dylan Murray, so keep that in mind. That indeed the ASOs do hit oligodendrocytes, that's very important, and that oligodendrocytes do express ataxin 3 and do have signs of aggregation-prone protein there. So you're right that the gene silencing approaches are going to have to hit non-neuronal cells as well. To the extent that the problem is a maturation problem in oligodendrocytes, that may be happening so early in the course of a human life that we are not actually able to target that. So, one thing we recognize that these diseases start to have symptoms at 20 or 30 or 40 years of age, but there might be neurodevelopmental processes that set in motion things that we can't reverse. So that is a cautionary tale.
So I assume this is similar for all the other dementia, right? For Alzheimer's disease. Once someone comes with the symptoms, it's pretty much.
Let me say with the polyglutamine disorders in Huntington's disease, it's very clear that individuals who have expansion in Huntington, and that's a neurodevelopmental gene to begin with, have changes in brain development. Kids who harbor the Huntington mutation, who don't have disease but harbor the mutation, have changed brains. The brains are different. I think there'll probably not only be changes in the circuitry of the brain, but probably in some of the epigenetic modifications that exist within the genome too. I am late for another meeting, so I got to go, but I'm so happy I could participate, Magda, and I'm so happy I could hear Dylan Murray's talk as well.
Hank, there are two questions.
Oh, there are. Okay.
Do you mind just answering? I'm sorry holding you. The first one is it's in the Q&A. First one is from Han.
Yeah. Do you know which factors play a more important role in Huntington's disease? The repeat RNA, CAG, or the polyglutamine protein? Should we focus more on polyQ or CAG to treat the disease? It's a really good question from Hong Yuan. And if you talked to Jim Gazella and Marcy McDonald at Harvard, they would say that we're missing the boat. If we're focusing on the protein, we're missing the boat that really it's a CAG repeat issue, and that might be both epigenetic changes as well as RNA-based toxicity, and that expansions in the RNA itself may play a major role. There's a lot of controversy about which is potentially more important. My bias is that they are both important, but that we need to also be thinking about the protein as well. The antisense approaches will be eliminating the RNA target. So that's important to recognize. The approach of an RNase-based ASO will hit the nuclear RNA and reduce that as well. But I think it's highly relevant we need to be thinking about the RNA as well. Most of the studies that have looked at RNA toxicity in CAG polyglutamine disease have used very large expansions, and we don't know if they are supraphysiological and that we're seeing things that aren't real in humans. Christian Greisinger asked the question, when you apply the antisense RNA, how do you do that technically? These are directed directly into the intrathecal space. In our case, there's an cerebroventricular injection into one ventricle, and that gets throughout the brain and into the spinal cord. In humans, when they're given to people, they're given through the intrathecal space in the lower spine. Another question is, the start of your talk, you had differently long polyQ glues, but the onset seemed to correlate inversely with the onset. Did I get that right? The longer the repeat, the earlier the onset of disease. The longer the repeat, the earlier the onset of disease. And there's some data that in ataxin 1, SCA1, when you have a histidine that's in the middle of the CAG, it's also a change in the CAG, that you actually reduce the severity of that glutamine stretch. So it does suggest that it has to be an uninterrupted stretch of polyglutamine, and the people who look at the RNA would say an uninterrupted stretch of CAG. So it's a good question.